Welcome to the Department of Medicine Grand Rounds. Today it's my great pleasure to introduce Dr. John Stamatoyannopoulos. Dr. Stamatoyannopoulos, I've been practicing that all night. Received his undergraduate degrees in symbolic systems and biological sciences as well as plastics at Stanford before coming here to the University of Washington Medical School. Completed his internship and residency in internal medicine and Epigenomics, his fellowship at oncology and hematology at Dana-Farber, before returning here to the University of Washington, where he's currently an associate professor in the departments of medicine and genome sciences. He's received many awards and honors, been an active research mentor, and has published extensively in journals such as Nature, Science, and Cell. He directs the UW ENCODE project, a component of the NHGRI program to map all of the functional elements of the human genome. He also directs the Northwest Reference Epigenome Mapping Center, funded by the NIH Roadmap Epigenomics program, and oversees the High Throughput Genomics Center here at UW. We're thrilled to have him today to give his grand rounds entitled The Living Genome, Implications and Opportunities for Medicine. Please join me in welcoming Dr. Stamatoyannopoulos.
Thanks very much, Sam. It's great to be here. This grand rounds is going to be potentially somewhat atypical. There's not going to be a case presentation. And I am going to focus on something. It's probable that you have had a number of speakers come through over time which have spoken around the general topic of connecting changes in the genome with clinical diseases, traits, etc. You're going to hear a little bit of the other side of the story to that. And I'm going to be focusing my talk on what I'm going to call the living genome. And I'm going to draw a distinction between the DNA sequence and, by extension, sequencing as a modality with assay of the genome as it exists in vivo. I guess this is probably the closest I'll come to a sort of a case. We have the case of the human body. And the question, of course, is how does one genome, which of course you have in every cell in your body, give rise to very diverse anatomical structures, and of course to all of the physiology that we study in medical school and apply on the wards. So what I'm going to talk about, and just to give you an overview, because I'm going to assume that there is not universal knowledge about the organization of the processes that allow a genome to turn into the body. I'm going to give an overview of that and kind of highlight the key areas that are now coming under study and are very rapidly penetrating their way into medicine. And by that I mean into clinical assays and ways of thinking about disease that are going to be meaningful both for the development of therapies, diagnostics, and treatments in the future. So in the beginning, I'm going to start with it's sort of like PGY1. You start with anatomy, physiology. So we'll start with a little bit of anatomy of the living genome. So if you take the genome and ultimately, if you look at a cell, the cell's got a nucleus, and the genome, the DNA of the genome, is packaged into a molecule called chromatin. Chromatin is kind of wound up inside the nucleus in very, very complex ways. And if we just sort of pulled out a chromatin fiber and looked at it, we'd see that it was punctuated by genes. Fairly rare along the chromosome for protein-coding genes, and there are also genes that just make RNAs. And of course, the genes do produce the RNA, which eventually makes the protein. And if we now unwound this molecule a bit further and took a closer look, we would see other features which are going to zero in on something that I'm going to be calling in this talk the epigenome. And this is the fact that this molecule chromatin into which the genome is packaged is composed of these little sort of spheroid structures, the nucleosomes, and this blue here is the DNA that's wound around them. And in addition to the genes, one of the other major sentinel features of the genome is the existence of these regions called regulatory DNA. And regulatory DNA is where little molecules called transcription factors actually directly read out the information that's encoded in the genome. So the genomic information is kind of read out in two dimensions. One is the transcription of that information to RNA, which is eventually then, the RNA is translated into proteins, et cetera. So the information is read out at that level. But there's a class of molecules that reads the information in the DNA directly. These are little proteins that recognize specific letter combinations and codes. So for example, this protein might recognize this combination of letters in approximately this proportion. So in other words, it likes to see G's more in that position. This one here may recognize a different letter combination. And there are over 1,000 of these proteins that exist, these transcription factors, that read out different combinations of the information in the genome. And the way the genome works functionally is that the packaging is so important and so tight into these nucleosome structures that it's very difficult for these proteins to get in. So they have to team up and come to regions of the genome which are a couple of hundred nucleotides long and essentially collectively attack and get in there and do their thing. And when they do that, several things happen. It turns out that what they can do is they can recruit other players into the game, and those other players can do things such as chemically modify the tails of these histones. They can also put on chemical modifications of the DNA itself. So all of these features, everything from the chromatin structure to the RNA to where these regulatory proteins are binding, to methylation to histone tails, all of these come under the rubric of epigenomic features, something that is kind of layered on the human genome sequence. And these now are under intensive study, as I will discuss a little bit. And more importantly, they are starting to become directly relevant for the understanding and treatment of disease in several ways. As I'll spend the bulk of the middle of the talk talking about, it turns out that changes in these regions are emerging as major players in mediating common diseases and also underlying the basis of common quantitative clinical traits, including exactly the kinds of traits that you measure every time you order any kind of blood test or whatever on a patient. These modifications that are put on to these nucleosomes here are put on by enzymes, which are now emerging one after the other out of studies of cancer genomes. And there's over 100 drugs that are in the pipeline currently that target the enzymes that put these kinds of modifications on DNA, many of which have been shown to extend patient survival in many cancer contexts. So these regions here, the regulatory regions, again, are kind of the key to all of these epigenomic processes. And without them, the genes really can't do anything. And these regulatory regions have a variety of different names according to what they actually do. So some of them are called promoters, and they sit right at the beginning of genes and sort of, drive the actual transcription of the RNA. But the majority of them actually fall into this class called enhancers. And these are things that are out there. I've sort of indicated there are lots of them around the genome. And what they do is actually control, or they're the major control vehicles for the gene. They're kind of the major on-off switches, and the promoters are sort of more the kind of the dials. And one of the key features that distinguishes the living genome from the static genome is, people show these pictures of the genome sequence, and they say if you stretch the genome out, it's a meter long, et cetera. But once you get the genome inside the cell, the topological orientation matters. So whereas the human genome sequence is straight, the living genome is very contorted. And the reason is that the way gene expression happens and gene regulation happens is these regions essentially reach over and touch the genes. And so in the nucleus, this is actually sort of sitting on here, and this is what's driving the expressions of genes. Now, what that means in a broader sense is that the genome is folded up very carefully in such a way that all of the right gene activity has to happen. And more importantly, when we try to determine and map features onto the human genome, mapping them onto the linear genome is one thing. But that linear genome has a three-dimensional instantiation that we have to understand. And there are now technologies that allow us to go in and assay that structure and to understand the connection between these on a very large scale. And even to make models of the genome. This is actually a stylized version. This is what's called a Hilbert curve. But it essentially indicates the idea that there could be two regions of genome which are physically extremely close to each other, but are on a linear molecule very, very far apart. Could be millions of base pairs apart. And this is sort of an actual model of how the genome is put together. So this principle, the genomic distance is not equal actual distance, turns out to be very important in understanding how the information coming out of genetic studies that are trying to inform us about the genic causes of disease actually work. So I'm going to draw a few distinctions here between the genome and the epigenome. And one of them I mentioned is that the genome is, of course, linear, and the epigenome is, in functional form, highly nonlinear. The genome is essentially fairly information poor. And by that I mean that at every position in the genome, there's basically one thing that can happen. There's going to be a single letter code, and that could be one of 4 bases. But overlying that, now, with the epigenome, you have a wide variety of different events that can overlie any given position in the genome, and we're still trying to understand how deep that rabbit hole goes. The genome is unidimensional, and by that I mean that you've got the same genome in every cell type in your body. The epigenome, by contrast, is essentially completely differently organized in every cell in the body, and in fact is the basis for that anatomical organization. The genome is temporally static. It doesn't change during your lifetime, except maybe acquiring a few mutations, cancer being sort of a special case. But the epigenome is tremendously dynamic, and it is dynamic not only on a scale of lifetimes, it is dynamic on a scale of minutes. As far as we can measure, it could even be a dynamic on a scale of seconds. And by that I mean that the kinds of changes that we can literally measure now that occur on the genome, if I expose a cell to a drug, et cetera, I can see changes on the epigenome in minutes. And that is something that is more characteristic, this sort of temporal dynamism is more characteristic of the kinds of tests that we do a fair amount of in memory. The epigenome also has one important and characteristic which has recently been recognized, but I think which is going to have a lot of implications, is that it has the capacity for memory. In other words, that you can have information essentially written on the epigenome in the form of chemical modifications, in the form of particular clusters of DNA binding proteins that hang around in response to prior events, such that you can detect signatures in the epigenome of cells and tissues having been exposed to something in the past. And that's a fairly new dimension which combined with technology, which inevitably is going to come around, is going to perhaps change perspective on how we monitor and look at a lot of things. Okay, so how do we understand and find features of the epigenome? How do we literally map information onto the epigenome? Or onto the genome that corresponds to the epigenome. So the idea is what we really want to do is, figure out where in the genome all of these kinds of things are happening, or all of these kinds of things are happening, or even RNA. So RNA is a pretty simple example. If you collect the RNA and you do DNA and you sequence it, you convert it to DNA and you sequence it, you can take that sequence and you can align it, map back to the genome, a fairly quick algorithm, and it can tell you basically all the bases in the genome that get turned into RNA. Well, it turns out that analogous methods over the last few years have been developed for mapping various of these other features, and each of them has kind of their own little tricks. There are special ways of mapping DNA methylation. There are ways of mapping these chemical modifications of histone tails, and the way that works essentially is by finding an antibody to those regions and essentially attaching the antibody here, carving everything up, and then just pulling down the protein stuck to the DNA, washing away the protein, taking the DNA, sequencing it, and mapping it back to the genome. These regions here, it turns out, have a very unique physical property, which was discovered in a variety of labs and really extensively developed by Harold Weintraub and Mark Gerdine here many years ago at the Hutch, and that is that these regions of the genome have a unique physical accessibility to molecules that enter the cell. So if we drop in a little enzyme that cuts DNA, it turns out that that enzyme can't cut anywhere in the living genome except these regions. And the canonical enzyme used for this is something called DNase 1. You may know this as a drug given to CF patients occasionally. But DNase 1 gets in there and cleaves the genome and really kind of actually vaporizes the DNA in these regions. It makes a little puff of stuff that comes out, and it turns out that you can collect those and sequence them and map them back to the genome, and each one of these little dots is sort of one of those sequences, and you get a big pile of tags that tell you where these regions are, and you can convert it into a little signal, and the result is that if you do this over and over for different cell types, you can make maps, and these maps now are available through web interfaces called genome browsers, and you can go and dial up a bunch of different cell types. You can pick the region of the genome you want, so showing a fairly well-known locus to any of the geneticists or lipid people. So this is about 100,000 base pairs of genome, and you can just literally track along and sort of read where these regions are. So you can, this is how you get maps of sort of where the regulatory DNA regions are, and thereby all the promoters, all of these enhancers that are controlling the gene expression, et cetera. But now if you went through, I'm only showing you 100 KB of genome, genome's big. So if you went around and you sort of counted up all these regions, you would find that in the average cell, there's somewhere between about 100,000 and about 250,000 of these regulatory DNA regions that are out there and they're active. That sounds like a lot, but it's only about, in aggregate, since each is quite small, it's only about 1% of the genome on average. So it's a compartment roughly the size of the total compartment that's giving rise to proteins. Now, the challenge, of course, is that most of the genome, if all of these things were kind of in the same place in every cell, it would be a cakewalk. But the problem is most of the genome kind of looks like this, where you have things that are highly selective for particular cell types, or this kind of on-off patterning. And what that means is to construct very comprehensive maps of the epigenome, and this is true of any of the sort of epigenomic features. You really need to do very, very broad surveys. So you need to survey a wide variety of different cell types, tissues, structures, et cetera. And we have done so, and our colleagues in programs like the ENCODE project and the Roadmap Epigenomics project have done so for adult cells and tissues. And also here at the UW, we've had an extensive program to map the regulatory DNA and other features in fetal cells and tissues. And of course, you can do this in embryonic stem cells and various other kinds of things. And the basic result is after doing all of this mapping over hundreds of different cells and tissues, you do some accounting that sort of looks like this. Essentially, by the time you tally them all up, there are around maybe 50,000 genes, of which about 20,000 make protein, about 30,000 just make RNA that do various things. But it seems to be over 4 million of these kinds of regions around the genome, each of which is combinatorially activated in different cells to give rise to the human body plan, and also sort of underlying a lot of the problems that we see. So now all of this information exists. You can go to any region of the genome you want. You can pull up a public genome browser. You can put your microscope or magnifying glass in any region of the genome, pull up any cell types you want, and you can sort of have maps of where all this stuff is. And we had, along with others, had described several major aspects of the regulatory genome in a series of publications last fall, sort of the general lay of the land, the fact that we now have technologies that can see in all the way to see individual DNA binding proteins, that you can combine this information to make networks that are sort of complex computational understandings of how the cell is functioning like a little machine. And it turns out actually that those transcription factors are wired together very similarly the way neurons are in the brain. And also what I'm going to spend some time talking about is taking all of that information and crossing it with a parallel stream which has developed over the last 10 or 12 years in genetics and genomics, which is the quest to identify genetic variants in the human genome that are associated with common diseases and traits. So in approaching this problem, one has to think of a general principle, which is true of all aspects of genetic variation. And that is that all genetic variation is ultimately interpreted in an epigenetic context. By that I mean, if you have a variant that is in a, even if you have a variant that is affecting the amino acids of a protein, that variant is not doing anything until there is an epigenetic context that allows that gene to be expressed. On the other hand, variants that affect the regulatory DNA. So every one of those proteins now recognizes a little sequence, and if a variant scrambles one of the words, the protein can't bind, and then problems can ensue. And it turns out that our ability to assess the effect right now, here we're in 2013, of genetic variation in coding sequences and protein coding sequences is relatively poor, in the sense that we have algorithms that can make predictions about whether changes in a protein are important or not, but there are very few ways to test those directly. By converse, if a variant affects regulatory DNA, there are ways now. I mean, it does something by a very simple mechanism. It disrupts the binding of one of these little proteins. And it turns out you can measure that very accurately, so we can generally tell if a variant is impacting regulatory DNA. So before I look into this a little further, one of the key features of most of the traits that I'll call medical traits, and these are the traits that you encounter in everything from the clinic to patients and inpatients to whatever, are essentially quantitatively or continuously distributed traits. They're not binary traits that you can just see this in simple morphological traits, things you measure, blood pressure is continuous, all these things. And in general, this kind of distribution of features is not something that you're going to expect is going to be due to drastic changes in a particular protein or not, which tend to give you sort of yes or no type of phenotypes. And one of the paradigms that has been developed for trying to understand and identify genetic variation that's associated with these kinds of continuous traits, as well as common diseases, which in many cases have a wide and continuous spectrum, is the so-called genome-wide association study. And genome-wide association studies have the following setup. In case you're not familiar, it's actually fairly straightforward. You get a bunch of cases with the disease or trait that you're interested in, you collect a bunch of normal controls, you get the DNA, and you genotype them. Meaning what you do is you read out the genetic code at about either 500,000 to 1 million specific positions in the genome that are pre-chosen in the case of most of these studies because they are common variants. In other words, that there's variability at those positions in more than around 5% of the population. And then what you can do is that you have everybody's genotype at every position, and then you just run through the list. And for every one of these variants, you just have it to calculate a p-value. Is this variant associated with the phenotype or not? And then if you take those p-values and those variants and you plot them onto the genome, you get plots that sort of look like this, which is, all the chromosomes are down here, it's like the whole genome. And then you see individual, this is the p-values, and you see individual SNPs or regions that are shooting up like this. And these are the regions then where you have specific variants that are associated with the disease or trait you're interested in. And if you zoom in on one of these regions, for example, you can find, you typically find in about 80% of cases, you can find a single lead SNP that just pops up and says, aha, I'm the most associated variant in this region. So we now have quite a lot of these lead SNPs. And in fact, there have been over 1,000 of these genome-wide association or GWAS studies, and they cover just about every kind of disease and quantitative trait you can think of, from morphological traits, drug response, cancer, immunological disease, etc. And collectively, all of these put together have identified about 6,000 different variants that came off as these sort of lead SNPs. And if you look at those and you map them onto the genome, you find that about 5% of them land in the protein coding regions of genes, and then the rest are out there in the non-coding space, some of which, this is 100,000 base pairs to 1 million base pairs, some of which are quite far away from from any genes. And the name of the game here, ultimately, is not find the variant. It's actually find the gene, because ultimately when we look at physiological processes, et cetera, it's going to come down to a protein that's going to be out there and doing something. But so then the challenge becomes, how do we connect all of this variation that lands in non-coding regions with genes? Well, first of all, let's ask where it is. So if you take all of these variants here in the gray zone, and then you merge them with maps of regulatory DNA, you come to a very simple conclusion. And that is that the disease-associated variation is actually concentrated in the regulatory DNA. And if you look at it on a case-by-case basis, but again, sort of summarizing all of the different kinds of diseases and squishing them all together, you find that if those lead SNPs, actually about almost 80% are either directly landing in the regulatory DNA or are so closely genetically linked to it that we can't discriminate whether they're in it or not, but we presume them to be. But if you look at this direct hit category here, again, this is taking all comers, but as you know, with any kind of study, there are the good ones and there are the bad ones. And if you look at the ones that are unreplicated, this number goes a little down, but if you look at the ones that have identified associations with a disease or trait and a variant in which somebody else has gone, taken another population, done the assay, and replicated it, you find that this number now is climbing to almost 70%, with a cumulative accounting of about 85%. And in every dimension you look, if you look for stronger p-values, the numbers go up. If you look for bigger sample sizes, the numbers go up. So everything is trending and pointing towards the variants that are landing in regulatory DNA. Okay, now what I told you a few minutes ago is that the regulatory DNA is, although there's a lot of it out there, that only essentially a small amount of it, 100,000 out of that 4 million or so, is on in any given cell type where it accounts for a very small percentage of the genome. So the real question, though, is not are you landing in regulatory DNA, but are you landing in regulatory DNA that is on in the right kinds of cell types that are important for a disease or trait? And the basic answer is that that's what we see. So, here's a variant associated with QRS duration on EKG. It lands in regulatory DNA that's on in the heart, but not in other cell types. Here's a variant associated with prostate cancer. It lands in a site that we see in prostate cancer cells, but not in other sites. Here's a variant associated with attention deficit disorder. It lands in regulatory DNA that's on in the brain, but not in other cell types. And it turns out that this is an incredibly systematic finding that you see across the entire spectrum of diseases and traits. And this is also just looking at the lead SNPs. It turns out that if you look at the entire spectrum of the data that you get from one of these association studies, you find that the effect is even more striking. So, for example, here's a genome-wide association study recently published in Nature, in which somebody took, one of probably the most commonly seen things on the wards, which are, results of a CBC, and took all the various parameters and did a genome-wide association study on a large number of individuals. And what I'm showing here is if you take the results of that study, and then this, down here you have all of these, there were like 1 million SNPs, and I've binned them into various different significances, so the more significant guys are over here. And then what I've looked at is taken the regulatory DNA from about 330 odd different cell and tissue types, and just asked, as a function of the significance of the association for these traits, how well do they localize within the regulatory DNA of a particular kind of cell? And it turns out that these are red blood cell traits. This red track right here are essentially erythroid cells, red blood cell precursors. The purple stuff is sort of any other white blood cells, and the gray is just about everything else in the body. So you can see that there's sort of a tremendous separation of the signal into the right cell types. So the epigenome, as I mentioned earlier, has a temporal aspect. And it has a capacity to remember, and it seems to have also a capacity to propagate itself. So that if we look at regulatory DNA that's active in the fetal stage of development, and so we have done now fairly extensive surveys from about the mid-first trimester to sort of the mid to late second trimester with a variety of different tissues. One can ask the question, well, we know that during gestation, you can have, the fetus is susceptible to various exposures. And we also know that there are epidemiological studies that connect various traits, blood pressure, BMI, cardiovascular disease, et cetera, with maternal exposures and maternal diet. And if you ask basically how much of this signal that we see of diseases mapping into the regulatory DNA is due to adult versus fetal regulatory DNA, you get the picture that actually most of it is coming out of regulatory DNA that is what I'll call fetal origin. This means regulatory DNA that starts out turning on in the fetus and then stays on in the adult, or regulatory DNA that is only on in the fetus and then shuts off. It turns out relatively little of it comes out in things that are purely adult stage, although there's certainly many things here that are present in the adult. But what you can do then is you can also separate and say, well, gee, what kind of, are there traits that have particular predispositions for localizing their signals in the fetal regulatory DNA versus the adult? And this is kind of important because in many ways, we don't actually know where the insult is occurring and where the risk lies for many diseases. So if you do this, you get the following picture. So these are the sort of disorders that are preferentially localizing variants in fetal regulatory DNA. These are the ones in the adult. And up at the top end here, what you have are sort of a little cascade of traits which are known to be associated from epidemiological studies with gestational exposures. You have these Barker traits, age at menarche, et cetera. At the opposite end of the spectrum, you have things that are associated with environmental triggers, asthma, and also things associated with aging, for example, Alzheimer's, breast cancer, bone mineral density, et cetera. Now, many of these, again, many of the of these disorders, we don't know and have not defined whether they have sort of fetal versus environmental components. And this is one of the insights that this kind of data can actually give you. So thus far, I've sort of presented the simple view of mapping everything to points in the genome. But as I mentioned, the real name of the game here is identifying the gene, trying to find the gene that's responsible for this. And to do this, we're going to essentially have to correct the genetic variation locations for the epigenetic circuitry. And essentially, one of the principles I had told you is that the regulatory DNA is typically controlling genes at a distance. Now, it turns out that those distances can be quite large. They can be tens of kilobases. They can be even megabases away. And again, giving rise to this sort of complexity. So what we have done is systematically, through a variety of methods, been able to compute how a significant fraction of the regulatory DNA is interacting with its target gene. And the basic result of this is that, so then essentially mapping elements to the genes they control, sometimes at great distance here. So this is a couple hundred kilobases. It turns out that the average regulatory DNA region is pretty precise in what it does, pretty simple function, and sort of connected to either one, about half the time to one gene, and the other time maybe to two or occasionally three genes. The average gene, however, is essentially receiving inputs from 15 to 20 different regions. So any given region is contributing just a little to what the gene is doing. But again, the magic here is trying to understand how do we connect the regulatory region to the gene. And as I mentioned, we have done that. And now when you start to layer this map of the connections of the regulatory DNA to the genes, you can start to correct the genetic association map for the physical map, or the epigenome map, as it were. And some interesting things come out of that. So for example, here's a variant, which I was just showing a moment ago, that this is a variant that's actually associated significantly with quantitative variation of platelet count that you see on a average CBC. It's sitting in this regulatory DNA region, but this region is actually controlling this gene over here, about 200 kilobases away, and it turns out this gene is involved in various aspects of platelet production and also in some myeloproliferative disorders and like essential thrombocytosis, et cetera. This is just showing that you can actually recover specific physical fragments that catch this guy here in the act of talking to this gene. Here's another example of a variant that's associated with various aspects of quantitative variation in white blood cell count. It lands in regulatory DNA. It's controlling, it's skipping over all these genes here, and it's controlling this gene over here about 300 kilobases away, which is the retinoic acid receptor alpha, which is involved in white blood cell production. Here's an example outside of the blood. Here's a variant associated with AFib. It lands in a regulatory DNA region that's specifically in the heart and not in other cells, not shown. And this region here is skipping over these nearby genes and controlling this little guy over here, which is the gene for atrial and natriuretic peptide. And this list goes on and on and on. And there are a couple of key features here. So firstly, what you can do is now correcting this map. Suddenly, all of these interesting genes start sprouting out of the woodwork. So for example, here's a variant associated with breast cancer, and it's in a regulatory region that's controlling a gene 411,000 base pairs away that happens to be a tumor suppressor that is known to be involved in mammary tissue. Here's a variant associated with rheumatoid arthritis that 360 kilobases away, its region is controlling a cysteine proteinase that's already been linked to articular erosion. So essentially the genetic information is pointing to one spot in the genome, but the regulatory information is essentially telling you where the signal is really, what the signal is really due to. This is kind of an overall picture of how far away on average are the genes that are being controlled by regulatory DNA regions that have genetic variants in them that are associated with a variety of common diseases and traits. And it turns out that over 40% of the time, that gene is over 250,000 base pairs away, which is several gene units, essentially, in the average genome interval. And the flip side of it is, you can also ask, well, what percentage of the time is the gene in the same genetic linkage block as the variant? And if you do that, the answer, and do that calculation, the answer is about 10%. So while resequencing those regions can help, in some cases, refine what the variant is, it doesn't at all essentially impact the issue that most of the action is going on far away. So I told you earlier that there are these little proteins, the transcription factors, and each of them reads a little alphabet, or a little code, a little word. And it turns out that if you look at these variants when they land in the regulatory DNA, that what they're doing is they're systematically landing in the recognition sites of one of these transcription factors. And the net result of it is that if in the wild type you see a signal that's, say, this tall, if you have a variant there, it actually impacts the magnitude of the signal. And what you can actually do with sequencing data is that if you have, for example, a heterozygous position, so an individual is heterozygous for a certain variant, with sequencing, you can take that signal we see, you can separate it and figure out, well, how much is coming from one allele versus the other allele, and then you can directly see the impact of a particular variant in a controlled environment on the DNA, and therefore flag that variant as a variant of functional effect. So, for example, here's a variant that occurs, and this is a recognition sequence for this kind of transcription factor. And if you have the T allele, which is the wild type, and then so the individual's heterozygous for that, they also have the G allele. Well, if you look at the signal, let's say in this site here, virtually all the signal is coming out of the T allele. It's just a little tiny thing coming out of the G allele. So the G allele is highly deleterious when you get it at that exact position. And you know, here's another example. So most of them are a little bit, you know, closer to this, maybe 2-to-1 type ratios, etc. And we have now cataloged over 100,000 such functional variants in the genome, and a significant proportion of them can be aligned with the genome-wide association data, such that when you have a variant that is associated with disease or trait and it lands in regulatory DNA, and we have adequate sequencing coverage to ascertain it, we find that nearly 40% of those variants actually have this property. So those data are pretty loaded with variants that actually have direct functional effect. So what about mapping this forward into physiology and also things that we can use that may change our opinion about how diseases work, how they're related to each other, et cetera. And you know, we have to admit that for many diseases, although we call it type 2 diabetes a disease, or we call certain inflammatory disorders a disease, we really are just doing, they're really sort of syndromes that are clusters of findings that we sort of treat equivalently, and we expect that there may be some different genetic basis for different forms. Moreover, we have certain different diseases, such as inflammatory diseases, that may have shared genetic liability. If you're at risk for this, you're also at risk for that. Well, one of the things that's great about regulatory DNA is that once you've got a variant in the coding sequence of a protein, sometimes you're kind of stuck, even knowing what it does. But with regulatory DNA, we can see, well, gee, it impacts this transcription factor, and this transcription factor talks to this one over here, and then we can actually systematically start walking up the chain and asking how all the information comes together. So in sort of a simple way, you can start with this guy in the middle here, which is a transcription factor called IRF9, and it's, if you look inside the sequences that it binds different places in the genome, you find there are variants associated with several different inflammatory disorders. But then if you say, well, okay, in the nucleus, who is this guy talking to? Well, it's talking to all these other factors and making this nice little network. And then you can ask, well, what are the kinds of diseases that we see mapping into the recognition sites of all these other factors? And it turns out it's just basically a who's who of all these different inflammatory disorders. Well, this transcription factor here turns out to be a member of a pathway called JAK-STAT, and JAK-STAT is important in type 1 interferon responses. These are things we have drugs for, but there are many diseases on this list which are not currently thought to be candidates for that kind of treatment, but maybe now, there is genetic evidence essentially pointing in that direction, albeit through the regulatory genome. You can also then ask, well, gee, are there shared features between different kinds of diseases? And so what you can do, for example, is go to a whole bunch of different autoimmune diseases. These are kind of all over the map. I mean, we normally don't, associate asthma with, with psoriasis or whatever. But it turns out that if you then ask, are these actually hitting sort of any common targets? It turns out there's a group of about 24 different transcription factors who turn up over and over again as targets of genetic variation associated with these diseases. And effectively, what it's showing is that there is a level of shared genetic liability and essentially shared risk for multiple diseases that is mediated at the level of the epigenome and the regulatory genome. You can construct these kind of maps for any set of diseases you want. Here's what a map of all the cancers that are out there. And again, it identifies a set of about 30 transcription factors in the middle that are repeatedly being hit. Many of these have known roles in cancer, many of them don't, and in fact, many of them have some interesting therapeutic applications. So one of the interesting features of disease is if you crack open, the pathology textbooks and you read about this and you look at the pathophysiology and do all kinds of other things, but it is the case that many of the disorders we look at, while we know their pathology, we're not exactly sure what exactly is the culprit cell type. In the case of inflammatory diseases, which one of these inflammatory cells is causing the action? In the case of many other diseases, what is the target tissue? What is the target organ for obesity? Is it the fat or is it the hypothalamus? And so what you can do is now you can take advantage of this breadth of accumulated and expanding regulatory DNA epigenome data to cross it with the disease data and then ask a simple question. Can we flag, using the genetic and regulatory data, can we flag the right cell or tissue that's causing the problem? And I showed you this graph earlier, but imagine that I had not told you what this was. Somebody had just handed me, said, I just did a genetic study. What am I finding here? Well, and we had these traits, but let's say we didn't know that those were regulatory traits. And what we would be able to deduce, red blood cell traits, what we'd be able to deduce right away is, in fact, that these traits are localizing themselves in red blood cells. Now, that's a fairly trivial example, but let's take a different, more complicated example, like Crohn's disease. And so Crohn's disease, the AV person's into. Got it, yeah. I'm assuming that's what that beeping is, but I'm not sure. Okay, so with Crohn's disease, fairly big cause of morbidity, certainly in in Seattle hospitals. And what we can do is we can do the same thing. We can take a genome-wide association study for this disease, and let's say we knew nothing about it. And what we could do is then just look at all the cell types and see where the variants are localizing. And the screaming signal that you get are this specific subclass of CD4 positive T cells, and secondarily this. And this is essentially what, you know, a couple of decades of a lot of other kind of experimentation has now pointed to. But it's something that just leaps out of these genetic association studies. And in many cases with quantitative traits, you know, things will localize directly into the target tissue. So here's a GWAS study that's done by Nana Sotodania and their colleagues looking at QRS duration, and you can see that this localizes really, you know, selectively into heart cells, essentially myocardial cells, even from the fetus. But what this actually shows is a very important feature. All of the stuff I've shown you so far in the sort of connection with the regulatory DNA has been based on variants that are in this bin right up here, because they attain what the statisticians like to call genome-wide significance, p-values of 10 to the -8. But what you can see is, so that's based on, you know, calculations made on genetic structure. But what you can see here is that there is a whole host of variants that are also selectively localizing in the regulatory DNA of heart, or in fact of blood cells, et cetera. So that cutoff's right about here for blood. There's a whole pool of variants that are out there that are doing things that we have not yet accounted for. And the basic message is that there are not single variants that are doing things. There are lots of variants. Probably they have small quantitative effects, turning the dial just a little bit from these regulatory DNA regions. But again, the key point is we need to map these to genes, and we need to expect that there's going to be a lot of different ways to break the system. So I'll just summarize some features of what I've just told you, and then give a perspective, I think, on where things are going. And this is for the roadmap Epigenomics project, but it's the same thing true for the ENCODE project, has created a fairly vast, high-quality atlas of the epigenomic states of normal cells and tissues. You can integrate this data to, you know, essentially identify important insights into cellular phenotypes and function. You've got this feature that the disease-associated variation is concentrated in regulatory DNA, which enables a very coherent approach. And one of the important things that I have sort of didn't make very clear is that everything I've shown you so far has been done out of reference maps that were created from normal cells. We're not looking at any pathological cells. However, when you combine it with the genetic data from pathologic or trait populations, you essentially have a very powerful tool for identifying the culprit genes. Essentially, this is really essentially, we are really at the beginning here. Although it seems like a lot has been done, a lot of things have been found, only a fraction of really the true diversity of human cell types and states has been covered. There are enormous challenges in looking at all of these data, but these things are things that are going to be solved. So where are things going right now? And how can you think of how genetics is, or how all of this is going to inform our understanding of genes and that are ultimately going to change therapies, diagnoses, et cetera? Well, I think that one of the key principles is that if you think of the gene and the genome as a clock, there are many, many ways to break the clock. And we should not be overly concerned with identifying the actual cause of every single change that we find out there that's associated with a disease. And so, for example, you know, with let's say Mendelian disorders like cystic fibrosis or whatever, if you go to a gene, you find that there are, in every case, tons and tons of many, many different mutations in the coding sequence that give rise to some defective protein and the phenotype. Our expectation is exactly the same in regulatory regions. But remember I said there's 15 to 20 of these that are feeding into every gene. So there's a lot of ways, a lot of gears that can break off, et cetera, that can happen. But the real key is pinpointing the gene, understanding the networks, the pathways, and how that feeds into physiology. And so the way you should sort of think of this kind of variation that's out there in distinction to variation in the protein coding sequences is essentially that, for common diseases and traits, what we expect is that most of the variation out there is going to be regulatory variation, budging that dial. There is going to be some obviously some protein coding variation, but on average, the effect of each variant is going to be very weak. And this contrasts quite significantly with the case of rare disorders with fairly dramatic phenotypes, Mendelian disorders, where it's predominantly protein coding variation, but actually there's a fairly large, or significant anyway, undefined regulatory component. I mean, 10% of CF is coding negative, and there are many other Mendelian disorders that we don't even know the numbers for. But I think in terms of, so this is in terms of generally understanding disease and putting things together. But there's another very practical aspect which is going to be accelerating in the next 4 to 5 years. And that is taking advantage of the fact that the epigenome is highly variable and, as I indicated earlier, is highly dynamic. And what this means is now that we can make measurements on a time scale that is actually the same time scale that we use for many other measurements in the clinic or in the hospital, that are dynamic parameters of the human body that we are not measuring. But you're measuring things in the blood or in other tissues that are going to give you direct readouts of the actions of things like drugs, et cetera. And this is really an area that's going to be enabled by a series of technologies that are miniaturizing and sort of formatting these epigenomic assays for application in clinical settings in cost-effective ways, and also by studies which are now just starting to understand questions such as the memory component. In other words, can we look at the blood and detect exposures? Currently, it's probably the case that we could detect if you had received steroids, for example, in somewhere in the near past, because it produces a characteristic change in the epigenome that even after the drug washes away, you can still detect. And who knows how many other things out there have that kind of property, but we're going to find out, and those are obviously many very potentially highly clinically useful features. So with that, I just want to close and acknowledge several of my very talented students and fellows, particularly Matt Morano and Shane Neff and Jeff Fierster and Andrew Strigakis and Tony Rabacek. This is our informatics group, without which we are dead in the water. Our team at the Epigenome Center, which is responsible for all the data generation, and a variety of collaborators here and abroad, and the funding for this largely comes from the NHGRI project and the Roadmap Epigenomics project from the NIH Common Fund. So with that, I'll stop and take questions.
In your maps where you show differences in the epigenome versus in the affected cells versus the uninfected cells, are those differences in expression, or is it actual differences in the genetic code? And if so, how do those occur in those cells and not in the other cells of uninfected tissues?
Do you mean, are you talking about differences in the epigenome between two different tissues, or between, let's say, liver and eye or something, or between?
The red cells that are affected, you showed the increased frequency of certain genes, and I'm not clear if that's the genetic code is different or just the expression of certain DNAs.
Let me explain. Let me explain this a little bit differently. What this is taking normal cells, in this case erythroid cells, and creating a map of where all the regulatory DNA is. So there's like 4 different kinds of these. so a couple 100,000 regions around the genome. And I'm doing the same for another huge number of cells, okay? So what we basically have are just the positions in the genome where this regulatory DNA is. Then if you take red blood cell traits, so for example, quantitative, so you take now a population of 10,000 people, and you take their CBCs, okay? And you genotype all those individuals. And then what you can do is for every single one of these parameters, what you can do is identify genetic variants in the genome that are associated strongly with the quantitative variation in that parameter. So then what you ask, what this is asking is, well, each of those has a significance. I have some variant that's weakly associated with this variation. I have some that are really strongly associated with the variation. And this is just asking, where are those variants in the genome, and are they landing in the regulatory DNA, but of what cell type? And it turns out that most of those variants in this case are localizing themselves specifically in the regulatory DNA of the red blood cells. So it's basically showing that the genetic study is concentrating its findings right exactly in the cell type that is essentially responsible for the finding.
So I have a number of questions, but I'll just ask one, because otherwise it'll take up another hour. The role of regulatory RNAs was not mentioned here. And I'm just wondering, I'm trying to figure out what the mode of interaction is between the two disparate elements that may be, a million bases apart. Is that the mode of interaction? Is it that your enhancer element or your regulatory DNA is encoding a small RNA or microRNA, or and I'm trying to get at why they need to be why you think they're closely associated in space.
Mm-hmm. Yeah, so there are RNAs out there that appear to soak up various regulatory proteins, and there have been some hypotheses that they may be involved in various aspects of genome function. Most of these connections, these long-range connections between the promoters and the enhancers, are mediated, I think, in part by two processes. First of all, the folding of the genome that passively brings regions into some proximity, and then also the building of transient protein bridges. So, for example, there's a protein called mediator, which can grab on one hand of the enhancer and grab on one hand of the promoter and bring them together. But it's not the case that these things are stuck together continuously. They're actually sort of coming together and then flowing apart. And so the way I like to think of gene expression is that gene is sort of like, the vaudeville act with the stick and the plate spinning, right? The enhancer comes along every now and then and just gives the plate a spin. But it has to stay in sort of the same spatial zone, so the genome stays approximately folded in the same way. But that's generally protein-protein interactions doing that.
So the dynamic features of the epigenome are information rich, but they also present a problem. Because if you want to study red cells, how do you do that? You take them out of the bone marrow, you culture them under certain conditions. How many cells do you need? Do you get one answer if you culture them one way and a completely different answer if you culture them another way? How much is that a problem for the kind of analysis that you do?
I think all of I think the answer depends on the cell type that you're looking at. Many of these, in the case where we have had an opportunity to compare ex vivo cells with cells that are propagated in cultures, that the epigenome is largely fairly stable. In fact, even surprisingly so in some cases. You can take, Th1 T cells and you can compare them to naive cells that are polarized to Th1 in vivo, and they're quite similar, or in vitro, sorry. But I think that the question is largely going to be obviated by the fact that there are now there's a trend in the technologies to be able to access the cells ex vivo directly. In other words, they'll be able to run the assays on small amounts of cells that are coming from BALs, from small biopsies, from blood, from you name it. And in fact, I think what we're going to find is that that the information that is sitting in blood is fairly tremendous in the sense that when it's circulating around, I think it's kind of picking up a lot of signals from other tissue beds that are potentially detectable in the peripheral blood. So I think that there's a wide range. Now, as you point out, It's dynamic. There are a lot of things that can measure. It's a fairly complicated space. But on the other hand, I think the machinery is now in place in terms of the research infrastructure that once an assay is available to try to translate those findings and really understand the clinical associations in a rational way.
One last question.
So in terms of nuclear structure, are there chromosome territories? And how dynamic are those? What regulates those?
Yeah, so there are territories of chromosomes, and what I'm talking about here is essentially this picture. Each of these little colors here is a different chromosome, and this is how they're folded up in the genome. And this is not a model. This is actually a computation off of actual data, interaction data. So chromosomes do segregate to particular areas of the nucleus. It is put together. We don't know a whole lot more than that at this point. I mean, we do know that it happens. And we also know that this structure is obviously completely lost when a cell undergoes mitosis, and the cell essentially has a way to magically reconstitute it fairly quickly after mitosis. But this is a large black box that exists currently.
Thank you very much.
Perfect talk. Thanks a ton. That was great.
Yeah, thanks. I didn't know.